Hello and welcome to Unacademy, a one-stop destination for all English medium UPSC aspirants. Welcome to the marathon session on science and technology. But before we start the session, a very important announcement. Mock tests are very essential before the UPSC Prelims. And I hope you all have appeared for the Unacademy All India Prelims mock test conducted on 26th May. And the next exam is on 29th. Don't miss the test. Let us start the discussion now. The first question. Read the statements. You have 60 seconds to go and let me know if you are done with the answer. Your time starts now. Okay, let us start the discussion. It is a very simple question. This is about the 5-layer protection. This is about the 5-layer protection on Delhi from all aerial threats. Okay, let it be a fighter jet, okay, or any air to surface missile, any kind of aerial threat. From that, a 5-layer protection, 1, 2, 3, 4, 5-layer protection. Okay, if the first layer fails, the second layer will protect. Okay, if the missile crosses the second layer also, then the third layer will protect. So that is the 5-layer defense mechanism. Okay, and the outermost layer is, in the 5-layer defense, the outermost layer is, okay, AAD and PAD. Okay, AAD, Advanced Air Defense, PAD, Prithvi Air Defense, AAD and PAD. The second layer is S400. Okay, this is something that we purchased from Russia. Okay, and this is something that made US unhappy about India because US wanted us to purchase Patriot Pack or THAAD instead of this S400. Okay, the third layer is Barak 8, the fourth layer is Akash, and the innermost layer is again indigenous BMD layer. Actually, this is one more move that again made the United States unhappy, because earlier we thought of purchasing, okay, the 5th layer from USA, but again India also had plans to acquire Integrated Air Defense Weapon System, IADWS, or the National Advanced Surface to Air Missile System 2, NASMAS 2. IADWS or NASMAS 2 from the United States. But later we changed our opinion and decided to make it indigenously. Okay, to use the innermost layer. Okay, but later it decided instead to use indigenously built ballistic missile defense system. Okay, so indigenously built ballistic missile defense system. BMD means ballistic missile defense system. Okay, right. So S400 and indigenous BMD, these are the two events India made USA a little unhappy, because like here they wanted us to purchase THAAD or Patriot Pack, and here, okay, they wanted us to take this NASMAS 2 or IADWS, but we have taken this from Russia and this one we decided to make it indigenously. Okay, so all the 5 are correct. Which of the above are not correct? None of the above are not correct. All are correct. None of the above not correct. It means all correct. So that is about the question number 1. Moving on to the question number 2. Consider the following statements. Okay, read the statements carefully You have approximately 30 to 40 seconds to go. Let us try to do it a little bit fast. Your time starts now. Okay, let us start the discussion. The correct answer for second question is C. Okay, 1 and 4 statements number which are true. Statement number 1 is correct, statement number 4 is correct. Okay, let us try to understand some basic concepts related to this and then come back to the question. Okay, we have two types of immune system. Okay, one is innate immune system. Innate immune system, and this is general immune system. Means what? Not specific to a particular pathogen. Not specific to a particular pathogen. Okay, like for example, when you cough, actually why do we cough? When we cough, there is a chance that the bacteria or the virus will be sent out of the body. Forcibly we are trying to push it out of the body. Same is the reason for sneeze. Mucus also, when the mucus comes out of the nose or throat, okay, then in the mucus a lot of virus or the bacteria will be sent out. It is a body's mechanism to send out the pathogen. Which pathogen are you sending? Are you specifically coughing out the coronavirus? Not possible. In general, any pathogens you have, you will do that. It means it is general, okay, to eliminate the pathogen from the body. So yes, this is matching with the definition of innate. Same way, increase in the body temperature. In general, when we get fever, we immediately get tensed and we will immediately try to take some paracetamol or something and will try to reduce the temperature. But it is not a right approach. In fact, fever is also a beautiful defense system of our body, because before we produce antibodies, the body temperature will be raised so that if the pathogen is heat sensitive, it will immediately die due to high temperature. That is the reason why we get fever. So, we should not immediately try to control the temperature. It is our natural body's mechanism to fight with the pathogen, okay, to kill heat sensitive pathogen. So, this is also correct. Next, this is related to T cells and this is related to antibodies. The T cells and antibodies, the second type of immune system is called as adaptive immune system. Adaptive immune system. Adaptive means what? Adjustable. Adaptive means adjustable. Okay, it means this is very specific. It means our body keep adjusting. For example, if you get cholera, then it will produce antibodies for cholera. Okay, next you will get typhoid, then you will produce antibodies for typhoid. After that you got some COVID-19. Okay, or a so unfortunate person got cholera, typhoid, COVID-19, everything. Okay, yes, in that case the person will start producing the COVID-19 antibodies. Okay, so that is how the body is getting adjusted to the type of infection the person is having and producing specific antibodies for it or specific T cells for that purpose. So they both comes under these two. Statement number 2 and 3 comes under adaptive immune system. Statement number 1 and 4 comes under, okay, this innate immune system. So therefore, which of the above are true with reference to innate immune system? 1 and 4 are correct. C is the right answer. Okay, moving on to the next question. Okay, the third question. Read the statements. You have approximately 40 seconds to go. Your time starts now. Okay, let us start the discussion. Okay, the right answer for this question is, okay, the right answer for the question is D, which are not true, none of the above. It means all are true. All the statements are true. Now let us give the explanation. Answer is different, explanation is different. You got the answer. Now let us see the explanation. This is not as simple as the first and second question. This is a little deeper, so complete focus here, please. Okay. First of all, this is something called as immunotherapy for cancer. Immunotherapy for cancer. And Tasuku Honjo and James Allison, they received Nobel Prize for this immunotherapy for cancer. And it is a factual point. There is nothing to explain in this, and this is correct. Tasuku Honjo and James P. Allison. These two people got Nobel Prize for this immunotherapy for cancer. That is correct. What is the topic we are discussing? Immunotherapy for cancer. Now we have to understand how does it work. What is this immunotherapy for cancer? Look at here. Focus on this. Okay. This is a T cell. T cell. I hope you remember that, okay, in general, T cells will kill cancer cells. Not only cancer cells, altered self cells. Altered self cells. We discussed about this, okay, in earlier class, yesterday or day before yesterday. I took the example of mera hai par teda hai, that one. Okay, altered self cells. Okay, cancer is, for a cancer patient, cancer is his own self cell, but altered because of the disease called cancer. So we need to kill that. It means in general we have to protect the self cells, but because they are self, we should not protect the cancer cells. That is the meaning of altered self cells. Okay, who will kill this altered self cells? T cells. It means T cells' duty is to kill the cancer cells. Is it clear? Okay, then they have two structures on them. Okay, keep remembering all the points, keep using the terms in your mind, or keep messaging me the terms that you are listening. They have two structures on them. What are they? PD1 and CTLA4. PD1 and CTLA4. Okay, what are they? They both are, okay, this PD1 and CTLA4 are brakes on T cell. What is the function of the brakes? They control the speed of the vehicle. Is it clear? They control the speed of the vehicle. Okay, it means they control the T cells. Okay, why to control the T cells? Because these T cells are killer cells, isn't it? T cells are killer cells, so they must be under the control. So who will control? Just keep messaging me. Who will control the T cells? PD1 and CTLA4. So what are they? They are the brakes on the T cells. Okay. For simple understanding, let us compare. Okay, let me compare. This is just a funny comparison. Okay, let us compare T cell with my pet dog. Okay, and let us compare these two, PD1, CTLA4, as leash. It means you are holding your dog with a leash. You are holding your dog with a leash. Okay, and you went for a walk. Nice, you are enjoying, your dog is also enjoying. Okay, but somebody tried to attack on you. Some enemy came. Somebody tried to attack on you. Then tell me what you will do. You are walking with your pet dog, holding the dog with a leash. Somebody tried to attack on you. What you will do? Unleash. So that the dog will go and attack on your enemy. Is it clear? So same way, this is like my pet dog. Okay. Can you guess who is my enemy here? Whom am I referring to as enemy here? Cancer cell is my enemy. As long as there are no cancer cells, hold your leash. But when the cancer cells develop, unleash it. so that the T cell will go and attack on that cancer cell. That's it. Okay, this is the immune therapy for cancer. I hope you understood what is the immune therapy. For this clever idea of unleashing the dog, James P. Allison and Tasuku Honjo got the Nobel Prize. But if you unleash your dog, you will not get any award. Don't do that. Okay, so here, okay, how to unleash? Our next question is how to unleash. What they have done? Okay, they produced antibodies for PD1 and CTLA4. They produced antibodies for PD1, CTLA4. In general, what the antibody will do? Antibodies will destroy the antigen. Is it clear? For example, polio antibody will destroy the polio antigen. PD1 antibody will destroy the PD1 antigen. CTLA4 antibody will destroy the CTLA4 antigen. So what they do? They destroy the PD1 and CTLA4. Now what happened? It is like a dog which is unleashed. It is like a vehicle whose brakes failed. Is it clear? Brakes failed. The car will go and attack on the other person, or this dog will go and attack on the other person. The other person here is cancer. So this is the story of immune therapy for cancer. Explanation completed. Let us check the statements. Okay. First statement, yes. Okay. Tasuku Honjo and James P. Allison got the Nobel Prize for this discovery. Okay. And the immunotherapy for cancer is by inhibiting the brakes that prevent the T cell from killing the cancers. They are holding the T cell. Is it clear? Like a leash. Now unleash it. Is it clear? The brakes are holding the car. Like cut the brake wire like that. Okay. Next. Okay. Pembrolizumab. Okay. This is a factual information. Nothing to explain. This is an antibody. I told you they produced antibodies for PD1, right? They produced antibody for PD1. Its brand name is pembrolizumab. I hope you got this. They produced antibody for PD1. That antibody brand name, just like paracetamol brand name Dolo 625, like that. Okay. So, a new drug called as pembrolizumab, an antibody with PD1 inhibitory capacity, means an antibody that can destroy the PD1, was approved by the FDA in the year 2014 for the treatment of melanoma. Melanoma is a type of skin cancer. Okay. So, yes, all the three statements are correct. The question is which are not true. There is no untrue statement. All are correct. This is the complete explanation for the question number 3. That's it. Moving on to the question number 4. Read the statements. 40 seconds to go. Your time starts now. Okay, let us proceed with the discussion. This is also a simple question. Fourth question, right answer is A. How many of the statements are correct? Only one statement is correct. Which statement is correct? The second statement is correct. The second statement is correct. Okay, let us try to understand what it is. First of all, okay, we have something called hemoglobin. Hemoglobin. Okay. Yes. All of you tell me what is the function of hemoglobin? It will carry oxygen and carbon dioxide. Okay. It will supply oxygen to all the body tissues and it will collect, okay, a carbon dioxide from the body tissues and releases it out. So that is hemoglobin. Is it clear? Then, in the hemoglobin, heme means iron. Listen carefully, heme means iron. Globin is the protein part of it. Protein part of hemoglobin is called as globin. This protein part will have 4 polypeptide chains. The protein part will have 4 polypeptide chains. Okay. What do you mean by polypeptide chains? I hope you all know that proteins are made up of amino acids, isn't it? Proteins are made of amino acids. Okay. And the bond between amino acids is called peptide bond. Peptide bond. Like for example, these are two, this is an amino acid, this is an amino acid and this is, okay, this is peptide bond. Is it clear? Therefore, proteins are also called as polypeptides. Polypeptides. Okay. In the hemoglobin, heme is iron part, globin is protein part. The protein part is composed of 4 polypeptide chains. What are they? 1, 2, alpha, alpha, 2 alpha and other 2 beta, beta. That is 2 beta. Total 4 polypeptide chains. So, this statement is wrong. The protein part of the globin is made up of 2 polypeptide chains alpha beta. Wrong. It should be 4. Okay. Then, now we have different types of hemoglobins. We have different types of, see, hemoglobins are ferroproteins. Okay. What do you mean by the word ferroprotein? Iron containing protein. Iron containing protein. Where do we have them? Red blood cells. Heme is a large organic molecule having an atom of iron. Do you also have this doubt? I had this doubt when I was a child. That is, why don't the blood rust? Okay, in the blood you have iron and there is a lot of water in the blood. So, why don't the blood rust? Is it clear? Because the iron in the blood is not free. The iron in blood, this is another fact, okay. Iron in the hemoglobin is inside a porphyrin ring. This is not a free iron, it is inside a porphyrin ring. That is the reason why. What is rusting? Rusting is oxidation of the iron. Oxidation means what? Addition of oxygen. You need a leftover valency for the oxygen to bind. But here there is no valence, like it is all occupied. It is in an enclosed ring called as a porphyrin ring. That is the reason why the blood will not rust. Okay, that is just a fun fact. Keep it aside. Okay, and don't ask me where is the fun in that. You have to assume like that. Okay, and iron in the hemoglobin is inside the porphyrin ring. And heme is a large molecule having 1 atom of iron. Okay, and the protein part is made up of 4 polypeptide chains, right? Then next point, we have different types of hemoglobins. Okay, we have different types of hemoglobins. By the way, today's class is not pure static, not pure current affairs. Kind of a mix. You can assume basics, but some current affairs are also there in this. Okay. Types of hemoglobins. Okay, we have adult hemoglobin. Don't think that we get adult hemoglobin after your 18th birthday. This adult is not that adult. Okay, so here adult hemoglobin will start getting produced the moment you are born. Okay, the moment you are born, the adult hemoglobin will be produced. But we also have fetal hemoglobin. Means, what is fetus? The baby still inside the womb, not yet born, completely formed human baby but not yet born, that will have fetal hemoglobin. And we also have embryonic hemoglobin. We also have embryonic hemoglobin. Means we have this type of hemoglobin when we are in the stage of an embryo. What is an embryo? It just looks like a group of cells. It does not look like a human baby. Just looks like a group of cells. Okay. In that stage we have another hemoglobin. By the way, leave this question. Did you ever wonder why do we have these many types of hemoglobins? Okay. Very interesting. Okay. Let us suppose a pregnant lady. Over enthusiastic. Okay. Went for mountain climbing. Who asked her to climb a mountain when she is a pregnant? Okay, went for a mountain climbing. Okay, what happened? Got oxygen scarcity. Oxygen scarcity. Okay, now there are two lives in her body. Is it clear? Her body plus the fetus inside. The fetus inside. Is it clear? Okay, but you have enough oxygen only for one person. You have enough oxygen. I told her not to go, but she went. Okay, you have enough oxygen only for one person. Okay, so you have to choose between the two, mother or baby. Is it clear? Okay, now tell me whom the nature will choose, mother or baby, whom the nature will choose. I mean, preference should be given to whom? Okay, no, fetus should get the preference. Fetus gets preference. Why? Not because mother can die. No, that mother also should not die. Fine. Okay. But mother is strong enough to bear the scarcity for some time. I hope you got the answer, Palak. Okay. Does not mean that the mother should die. But our singers are singing breathless songs. 4-5 minutes they are not breathing at all. So it's okay. 3-4 minutes oxygen does not get also. The mother can survive. So the baby is very delicate. The preference should given to baby. So this is the reason why we have special fetal hemoglobin which is having more affinity to oxygen. It means, let us suppose you have oxygen, adult hemoglobin, fetal hemoglobin, both are competing. From my discussion, you tell me what you understood. The oxygen preferably binds to adult hemoglobin or fetal hemoglobin. Yes, everybody. Okay, the oxygen, yes, wonderful. The oxygen preferably binds to fetal hemoglobin. So, did you see? The mother's love is extended to the biochemical level also. So, respect your mother. Okay, going back to the, discussion. So, we have different types of hemoglobin here. Okay. Adult hemoglobin composition is alpha 2 beta 2. Is it clear? Adult hemoglobin composition is alpha 2 beta 2. Fetal hemoglobin composition is alpha 2 gamma 2. Is it clear? Okay. See, alpha, alpha, beta, beta. This is HbA. Alpha, alpha, gamma, gamma. Okay. That is, HbF. So that is how we have different types of hemoglobins. Is it clear? Okay. But they both are normal hemoglobins. They both are normal hemoglobins. But here we have some abnormal hemoglobins. Is it clear? We have some abnormal hemoglobins. What are they? Hemoglobin S. Did you hear about sickle cell anemia? Hemoglobin S causes sickle cell anemia. Okay. Now, why do you get sickle cell anemia? What happens here? Okay. Beta 6 glutamic acid to valine. You may say, what is that, ma'am? Looks like Greek and Latin. So let us try to understand. Okay. Let us suppose 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. What are all these balls? What is this chain? Let us imagine this is beta chain. And what is each ball? Each ball is an amino acid. I told you that protein is a chain of amino acids, isn't it? Okay. So this is beta chain. Which beta chain? This one, the beta chain. Is it clear? Beta chain. I told you adult hemoglobin, HbA will have alpha 2 beta 2. Adult hemoglobin will have alpha 2 beta 2. In that beta chain, it is a polypeptide chain. It's a chain of amino acids. In the beta chain, 1, 2, 3, 4, 5, 6. Sixth position is originally glutamic acid. What is this glutamic acid? This is the name of an amino acid, okay? This is the name of an amino acid, okay? Originally in the sixth position you have glutamic acid, but that is changing into another amino acid called as valine. Is it clear? Then it is called HBS. But, okay, the same glutamic acid, if it gets converted to lysine, then it is called HBC. Is that clear for all of you? Yes? Okay. So, in the beta chain, the sixth position, original amino acid is glutamic acid. If that gets converted to valine, then it is called as HBS. If that gets converted to lysine, that is called as HBC. Okay. Then, they both are defective hemoglobins. Next, there is one more disease called thalassemia. Did you hear about thalassemia? Yes. Okay. In thalassemia, what happens is, okay, in thalassemia, what happens is, this is not a defective hemoglobin. Here, there are two types. Alpha thalassemia. Here what happens? Alpha chains are missing, total missing. Not a defective alpha chain, absence of alpha chain. Is it clear? There is another thing called as beta thalassemia. Here what happens? Beta chain missing. Not a defective beta chain, total beta chain gone, beta chain is missing. That is called beta thalassemia. Means here the hemoglobin will be only like this, beta missing. Is it clear? Here the hemoglobin will be only like this, beta. Okay, alpha is missing. You tell me which is more severe disease, sickle cell anemia or thalassemia? Sickle cell anemia, thalassemia, which is more severe disease? Yes, thalassemia, because in the sickle cell anemia you have beta chain, but one amino acid changed. But in thalassemia there is no beta chain, total beta chain gone. So that is the story of thalassemia. Is it clear? Now let us check this question. Okay. The protein part of hemoglobin is made up of 2 polypeptide chains. Wrong. It is made up of 4 polypeptide chains. Hemoglobin S involves a mutation of beta 6 from glutamic acid to valine. Correct. Hemoglobin C involves a mutation from beta 6 valine to lysine. Wrong. Okay. Beta 6 glutamic acid to lysine. Is it clear? Beta 6 glutamic acid to lysine. Okay. So, 1 and 3 are wrong, 2 is correct. Only one statement. How many is the question? So, therefore, only one statement. Second statement is correct. So, that is about the question number 4. Okay, now moving on to the question number 5. Okay, question number 5. Consider the following statements. Okay, consider. You have 40 seconds to consider. Your time starts now. If it is totally not known to you, take it as an opportunity to learn a new topic. Take it as we have approximately dealt with total more than 100 questions in the complete Conquer Prelims, but you are expected to get only 12 to 15 questions. Okay, so this is just an opportunity to learn. You don't know what is CAS-JV. Simple, it's an opportunity to learn what is CAS-JV. Take it like that. Okay. Okay, let us check the answer. Fifth question, the answer is, okay, B. And how many of the above statements are correct? Okay, correct. 2 statements are correct. Which 2 statements are correct? 2 and 3 are, okay, correct. Okay, 2 and 3 are correct. Okay, now let us leave the question for some time. Okay, and let us try to understand what is CAS-JV. Beautiful idea actually it is. Try to understand. Complete focus please. What is CAS-JV? Okay, now let us take sickle cell anemia. Keep responding. Sickle cell anemia or HBC or thalassemia. Tell me what is the problem in this? HbA is defective. Okay, is it clear till this point? No problem Dinesh, it's an opportunity to know. Okay, focus on this point. Don't focus on the point that you don't know this. Okay, sickle cell anemia, right? Or thalassemia. What is the problem? Adult hemoglobin is either defective or absent. Is it clear? If it is HbS or HbC, it is defective. If it is thalassemia, it is absent. Clear till now? Now, do you agree that something is better than nothing? Something is better than nothing. And take this like a flow chart. If point number 1 is not clear, you cannot understand point number 2. Okay, so don't miss anything in between. Okay, something is better than nothing. It means adult hemoglobin is defective. Okay, now normally answer this question. Normally, do an adult have fetal hemoglobin? Normally, adult will not produce fetal hemoglobin. Agreed? Yes. Now, why? Because a particular gene called as BCL11A gene, okay, BCL11A gene inhibits the production of HbF, that is fetal hemoglobin. Okay, I repeat, a person is having sickle cell anemia. So, there is a defective adult hemoglobin. Adult hemoglobin is not working. Okay, means there is no hemoglobin at all. And something is better than nothing. When you do not have adult hemoglobin, it is better to produce at least fetal hemoglobin. Do you agree with this? When you do not have adult hemoglobin, at least it is better to produce fetal hemoglobin. Actually, fetal hemoglobin is not suitable for the adult. But the best one is adult hemoglobin. But I don't have adult hemoglobin. So, why can't I at least have the fetal hemoglobin? Now, coming to the next point. That is, why don't an adult produce fetal hemoglobin? Fetal hemoglobin production is inhibited by BCL11A gene. Okay, now what you do? do gene therapy and cut down the BCL11A gene. Is it clear? Do gene therapy and cut BCL11A gene. I hope you got the point. BCL11A gene is cut. What is BCL11A gene doing? What is this gene doing? It is inhibiting the production of, just like the previous leash topic, BCL11A is like a leash for fetal hemoglobin. Now leave it. Nobody to hold the fetal hemoglobin production. So what happens? Fetal hemoglobin starts producing in the adult. So normally adult will not have fetal hemoglobin, but because here adult hemoglobin is not there, we are making the adult produce the fetal hemoglobin so that he will have something better than nothing. Is it clear? Okay, so that is called as Cas-JV. Why the name Cas-JV? Because we cut that BCL11A gene using CRISPR-Cas9. You all might have heard about CRISPR-Cas9, Cas. That is the reason why this is called as Cas-JV. I hope everybody understood this. It's a technique makes the body to synthesize embryonic hemoglobin in hemophilia thalassemia patients as they cannot produce functional adult hemoglobin. Okay, can you people identify where is the mistake? The exact wrong word in this sentence? Yes, everybody? The exact wrong word in this sentence is, very good, wonderful. Okay, Varsha. Embryonic hemoglobin. Total sentence is correct, but this is fetal hemoglobin. Very good. So, this statement is wrong. Cas-JV is a method of gene therapy? Yes, it is a method of gene therapy. We are cutting that. So, okay, what is gene therapy? Therapy means what? Treatment. Okay, giving treatment by dealing with the genes. Using genes to give the treatment is called as gene therapy. Is it clear? And Cas-JV utilizes stem cell technology. Yes, correct. Because we cut the BCL11A gene in the stem cells. In what cells we are cutting it? We are cutting that gene in the stem cells so that that correction will go to the other body cells also. So this is also a correct statement. I hope everybody understood this. Yes, the only wrong statement here is, okay, first statement is wrong. Okay, 2 and 3 are correct. Okay, yes. Moving on to the next question, that is Cas-JV. Next question. Question number 6. Simple question. 20 seconds to go. Okay, the correct answer is C. Okay, and this is the definition. What is phishing? What is phishing? Okay, right. This is when an attacker impersonate a legitimate entity. It means you will get a message from some HDFC bank, or you will get a message from Prime Minister's office, like that. Okay, such as bank or an organization. Okay, then you will think that it is a legitimate source. I got a message from HDFC, I have an account there, so I have to click on that. You will click on that and like a malware will be installed in your computer. Okay, so that kind of attack is called as phishing. Okay, the next question. The term used to describe a cybersecurity attack that interferes and alters communication between two parties without their knowledge is. 20 seconds to go. Okay, the correct answer is man in the middle attack. Okay, so sometimes what happens is a person comes in between or a software comes in between and will alter the communication between two parties. Okay, you will say yes, but the man in the middle will make it no, so that a wrong communication reaches the other person. That kind of a cyber security attack is called as man in the middle attack. Okay. Right, that is the seventh question. Okay, moving on to the next question, eighth question. Eighth question. Read the question carefully. 40 seconds to go. Your time starts now. Okay. The right answer is all the four. All these are the applications of this laser. Okay. But we have to know what is this laser. Okay. Let us try to understand this. The world's most powerful laser. It was a news in the Hindu newspaper. This was the title. The world's most powerful laser. A research center in Romania, part of European Union's Extreme Light Infrastructure project. As a part of this project, Extreme Light Infrastructure project, developed the world's most powerful laser. Who developed the most powerful laser? Romania. As a part of their Extreme Light Infrastructure project, developed this world's most powerful laser. Okay. A French company Thales, a global leader in aerospace, defense and security solutions, operates this laser. This company is currently operating using this particular laser. Okay. And the technology used in this laser is called as chirped pulse amplification technology, CPA technology. Chirped pulse amplification technology. This is a technique used to create ultra short. Okay, like in one of the earlier classes we have seen like attosecond light pulses, femtosecond light pulses, something like that. Okay, ultra short duration fast, okay, yet extremely high energy laser pulses, flashes. Okay, so what is chirped pulse active amplification technology? Ultra short duration flash, but very powerful, world's most powerful. Okay, in that one, that attosecond Nobel Prize, 2023 Nobel Prize Physics, what is the advantage? Powerful is not the 2023 Nobel Prize. Okay, the speciality there is shortest duration. Attosecond, that is 10 to the power of minus 18 of a second. That is the plus point. But here, the chirped pulse amplification technique, here the plus point is not, okay, duration. Here the plus point is powerful, intensity. So, this is speciality is speed. Here the speciality is power. This is also fast, but not attosecond fast, not that much fast. Here the speciality is its intensity. I hope you understood the difference between the two. Okay, this is called chirped pulse amplification technique. Short duration but extremely powerful laser pulses. Is it clear? Okay, and this scientist, okay, invented this particular technique. Okay, so they are awarded, these two scientists awarded the Nobel Prize in Physics for 2018. So this is the difference between 2018 Nobel Prize. What is that? Powerful technique. 2023 Nobel Prize, fastest pulse. Is it clear? Okay. For their groundbreaking work in harnessing the power of laser for advanced precision instruments. Okay. This is about this most powerful laser. What are the applications of it? Okay. Nuclear waste treatment. You can use this most powerful laser for treating the nuclear waste. How? Okay, when the nuclear waste is exposed to such high intensity light, it will undergo disintegration. Okay, the laser could be used to reduce the radioactivity duration of the nuclear waste, making its disposal safer and more manageable. It means you have nuclear waste. If this nuclear waste is treated with this laser, then the radioactivity of that waste will reduce. You can safely dispose this nuclear waste. That is one application. Next one, space debris removal. In yesterday or day before yesterday class, we have seen about the space debris or space junk. It's a huge problem and it may hit our satellites and destroy them, or it can harm our astronauts. Is it clear? And yes, okay, as Abhinash is saying, it will cause Kessler syndrome. What is this Kessler syndrome? Poem, Dinesh, very true, correct. Poem 3. Okay, so what is this Kessler syndrome? Kessler was, Donald Kessler, I think, was a NASA scientist. He predicted that, okay, one space debris will hit another object, then it will break down into multiple objects. Now the 4 will hit 4 objects, they will become 16. The 16 will hit 16 objects. So like a chain reaction, it goes on and it will make the space activities impossible for future generations. This is what Kessler predicted. That is called Kessler syndrome or the Kessler effect. That is the problem with space debris. And in my class at least, don't focus only on the question, whether the question is easy, difficult, you could do it, you could not do that. No. The terms used in the question, the concepts given in the question. I tried to cover, as a part of this Conquer Prelims, I tried to cover all the important news that appeared in the last year, right? Okay, so now space debris removal. Okay, with the increasing number of debris accumulating in the space, the laser technology could be employed. Very important, because, okay, we cannot use weapons in the space. Is it clear? So if you have to clear the debris, you have to clear like this only. Okay, could be employed to clean up the orbital environment, mitigating the risk of collisions with satellites and spacecrafts. It means if you put this laser on that debris, the debris will get disintegrated. Okay, if you put these flashes onto the space debris will get disintegrated. That is one advantage. Medical advancement. The precise and powerful nature of the laser could lead to breakthroughs in medical treatment such as targeted cancer therapy. See, if you put this light pulses onto the space debris, that space debris itself will get disintegrated. Nuclear waste will get disintegrated. After all, cancer cell, if you put these flashes on that cancer cell, the cancer cell also will die. So it's a good cancer treatment, cancer therapy and advanced surgical technique. You can do laser surgery without using the knife. The laser beam can be used to make the incision. Is it clear? Okay. Then manufacturing glass for cell phone screens and all. How? To cut the glass. Like evenly cutting. Okay. That line is not even, it is going wherever it wants, but to evenly cut the glass, we can use this laser beams. So these are the applications of this laser beam and all are correct statements. Is it clear? High precision, straight cut. if you cut the glass with a knife or something, it cannot cut the glass very properly, right? That is the reason why we can use it. That is question number 8. Okay. Moving on to the question number 9. Read the question carefully. 40 seconds to go. Suspense, Ishika. Okay, 9th question. A is the right answer. Okay. Let us leave the question for some time and discuss the concepts used in the statements. So, this question is about genomics. Okay. What is genomics? Okay. And what is genome? Okay. Genome is, okay, no, Manish, this is faster because the questions are simple. Okay, but it should not be the simple questions always. Some questions have a lot of deep concepts. So we had to spend some time to understand those concepts. These are, see, fishing, man in between, you know everything, like what to explain in that. Okay, so it all depends on the type of the questions. Is it clear? Okay, so first we have to understand what is the genome, right? Okay, then genome is one set of all chromosomes. One set of all chromosomes in, yes, try to fill the blank. One set of all the chromosomes in a cell or in the complete body? One set of all the chromosomes in a cell or in the complete body? Very good. In one cell. Okay. Then genome is equal to 23 chromosomes. Genome is equal to 23 chromosomes. Okay. Then you may ask me, no ma'am, in one cell you have 46 chromosomes. Okay. That is 23 pairs. Okay, or 46 chromosomes you have. But, okay, actually 23 chromosomes will come from the mother and the other 23 chromosomes come from the father. We have 2 copies of each chromosome. Is it clear? We have 2 copies of each chromosome, right? In one somatic cell, yes. Okay, so this is, so one genome is equal to how many chromosomes? One genome is equal to how many chromosomes? Everybody? 23 chromosomes. That is one genome, not 46. 46 is 2 genomes. One genome coming from the mother plus one genome coming from the father is equal to 46 chromosomes. Okay. Can you tell me one cell contains, okay, how many genomes? One cell contain how many genomes? Wonderful. 2 genomes. Is it clear? 2 genomes, because 23 will make up one genome. We have total 46 chromosomes. 23 together is one genome. The other 23 from the father is another genome. So you have one genome from the mother, one genome from the father. You have 2 genomes in one cell. Is it clear? Clear for all of you? So that is a genome. And what do you mean by genome studies? Genome studies or genomics. What do you mean by genome studies or genomics? This is, okay, like determining means what? Knowing the, okay, nucleotide sequence, knowing the nucleotide sequence of the genome. You all know that our chromosomes are made up of nucleotides called A, T, G, C, adenine, thymine, guanine, cytosine. Their sequence, determining or knowing the nucleotide sequence of our genome is called as genomics or genome studies or human genome project. Is that clear? Okay. We have different human genome projects. First we have Human Genome Project. This was completed in the year 2003 and it is a worldwide project. Okay, it means in that project they sequenced the genomes of the people from the entire world, not one country. They have taken the human beings from the entire world. Why to take these many human beings? Okay, answer this question. Do we both have same genome sequence? The nucleotide sequence of my genome, is it same as the nucleotide sequence of your genome? No. If that is true, okay, then there is no difference between you and me. It means we all have a different sequence of nucleotides in our genome. So, it is not enough if I know the nucleotide sequence of only my genome. Is it clear? So, what they will do? They will take lakhs of people from across the globe and they study the genome sequence of all of them. That is called as Human Genome Project 2003. Next you have, okay, Genome Asia. If you observe, I am just ignoring the question and trying to give you more information. None of these are covered in the question. The question is very simple. I have already given the answer, isn't it? We are trying to recollect some other information. Okay. So, Genome Asia 100K project. What is this? 1 lakh people, genomes sequenced. Okay, this 1 lakh people from only Asia, not worldwide, only Asia. In this, okay, 50,000 only from India. Is it clear? In that 1 lakh 50,000 samples taken only from India. All these people's genomes sequenced. That is Genome Asia 100K. Then you have other projects like Genome India project, Indigen project. So, as the name itself is explaining, here the sample taken from only Indians. How many Indians? 10,000 Indians. Did you understand this? 10,000 Indians. Is it clear? The Department of Biotechnology announced the completion of 10,000 genome project, an attempt to create a reference database of whole genome sequence out of India. Okay, fine. Okay, this is India. What other countries have similar projects? Okay, United Kingdom, China, United States. These are the other countries, okay, that have such programs to sequence at least 1 lakh of their genomes. India, USA, UK and China. These are the countries that are sequencing at least 1 lakh genomes of their population. Is it clear? Okay, then about 20 institutions across India are involved in this project. In this 10,000 genome project, approximately 20 institutes are involved, and the Indian Institute of Bangalore, Indian Institute of Science Bangalore, okay, and the Center of Cellular and Molecular Biology from Hyderabad. See, I came from Hyderabad and Bangalore. These two institutes are the lead institutions of this project. Okay, right. Okay, so this is about the 10,000 genome project. Okay, but not only this. Should we sequence the genomes of only human beings? No. Okay, we have to sequence the genomes of, okay, SARS-CoV-2. Why? First one, to make vaccines like mRNA vaccines. See, if you do not know their genomes, okay, how do you prepare DNA vaccines for them? Okay, to make DNA vaccines. World's first DNA vaccine for COVID-19, okay, is by Zycov-D. Okay, the world's first DNA vaccine is Zycov-D, made by a company called Zydus Cadila in India. India made the world's first DNA vaccine for coronavirus. Same way, okay, for diagnostics. Do you know PCR test? RT-PCR test? Okay, what is PCR test? This is identifying the coronavirus RNA in our sample. In the human sample, if you have the coronavirus RNA, it means you are infected by the coronavirus. For that purpose also you have to know the genome sequence of that. If you don't know the genome sequence of coronavirus, how do you identify that it is coronavirus genome? You will think that it is some poliovirus genome, right? So that is the reason why you have to do the genomics of this one also. Also to study the new variants. New variants. Yesterday we discussed about the flirt variants. Is it clear? That is evolution of the virus. That is the reason why we have to do the genome studies on all the pathogens. Flu virus, cholera, TB, HIV, Zika, Nipah, Ebola. We need to know the genome sequences of all the pathogens. Is it clear? Only pathogens? No. Okay. We are also doing the, okay, Earth Biogenome Project. Okay. Earth Biogenome Project. So this is a never-ending project. In this, the target is sequencing the genomes of every living organism on the earth. Okay. Okay, it will never be completed, but no problem. The target is not to complete the project. The target is to know more about more organisms. Every day, our knowledge about more organisms keeps on increasing. Biodiversity applications. This is all about genomics. Is it clear? Okay. Now moving on to the question. So, okay, question is of secondary priority. Try to understand the concepts used in the question. Okay, right. The precision medicine or designer drug. Interesting. What is precision medicine or designer drug? Okay. Designing a special medicine only for you. Okay. A designer drug for Bindu. Like that. Okay. This medicine will not work for anybody else. It will work only for this person. That is called precision medicine. Okay. If I want to design an exclusive drug for Priya Kumari, then I have to know her genome sequence, isn't it? Okay. So for that reason also genome studies are very important. Is it clear? Okay. Precision medicine, designer drug for particular individual. I need to know the genome sequence of that person. Okay. Okay. Then the next one. In gene editing, what is gene editing? Gene therapy. Just previously 5 minutes back we have seen BCL11A gene was cut. To cut the BCL11A gene, I have to know where is that BCL11A gene. For that I have to need knowledge about the, okay, complete genome. Is it clear? We have seen to cut the BCL11A gene. Okay, I will cut the BCL11A gene. Okay, but I should know where is BCL11. By mistake, if I cut the hemoglobin gene, person gone. Okay, forget about gene therapy. With extreme anemia, the person will die. So that is the reason why we have to know the knowledge about the genome, right? In studying differential effects of chronic infectious diseases in different individuals. Okay, see, did you wonder in the COVID-19 pandemic, some people died due to the COVID-19, but some people not even called a cold or cough, no symptoms at all. Is it clear? Why so much of difference? Why this much of difference? Why, like, it is not about the virus is dangerous. If the virus is dangerous, okay, if the virus is dangerous, everybody should die. Is it clear? No, not fear. Okay, many strong people died and many people who are good at fearing, they survived, not due to the fear. What is the reason? The reason is differences in the genome sequences. Is it clear? The difference in the genome, your genome is different, my genome is different. So, okay, my genome made my immune system stronger, so I could easily survive the pandemic. Is it clear? So that is how to study the differential effect of chronic and infectious. Some people will survive for decades with diabetes. Some people will die within 2-3 years after getting diabetes. Why chronic? Some people easily succeed the cancer. Okay, but some people, okay, may get succumbed to the cancer. So why cancer, diabetes, infections are affecting different people differently? Because their genomes are different. To understand that, we have to study their genomes. Next one, cloning and three-parent baby. Cloning means what? Copy. For example, I want to clone Einstein. Okay, means a person who is exactly like Einstein. And three-parent baby, a baby who will get genetic material from 3 genetic donors. Is it clear? Okay, so very briefly to tell you this, 23 chromosomes from the mother, 23 chromosomes from the father, and mitochondrial DNA. Mitochondrial DNA from a donor, because mother's mitochondrial DNA is defective. That is called three-parent baby. Okay, right. So, for this also we need genome studies. For that also we need genome studies. But if you have 1, 2, 3, 4 option, then you can tick on that. Did you understand? I kept this particular question like this because I just wanted to remind you one more time that multiple choice is all about choosing the most appropriate answer, not the correct answer, most appropriate answer. In fact, 1, 2, 3, 4 is correct, but 1, 2, 3, 4 is not there. Okay, so definitely cloning and three-parent baby, okay, require the genome knowledge to a lesser extent. You don't know completely about the genome, still you can do cloning and three-parent baby. But you cannot cut BCL11A gene without knowing about the genome. You cannot identify the pathogen without knowing its genome sequence. Is it clear? So they also require knowledge about genome, but to a lesser extent. So therefore the right answer is 1, 2 and 3. Is it clear? Okay, right. So moving on to the 10th question. This is about sodium ion batteries. Your time starts now. Okay. So the right answer for the 10th question is B. B is the right answer. Okay, 10B. 1, 3, 4. This is wrong. Okay. Okay, now leave the question. Let us try to understand what is a sodium ion battery, what is a lithium ion battery. Okay, let us go into the basics of that. Okay, then we'll again go to the question. Okay, right. Okay, sodium ion battery. Sodium ion batteries are the rechargeable batteries which require the sodium ion movement between the electrodes. Okay, during charging and discharging. Look at here. Okay, you tell me, during charging, when you are charging, look at the arrow mark. Okay, tell me the sodium ion is moving from positive to negative or negative to positive? During charging, the sodium ion is moving from, okay, very good Abhilasha, positive to negative. This is the arrow mark. During discharge, the sodium ion is moving, okay, from the negative electrode to positive electrode. These are the rechargeable batteries. Is it clear? These rechargeable batteries we use in EVs, electronic vehicles, okay, cell phones, okay, laptops, okay, and any battery like any battery that bikes, that is electronic vehicles, bikes, cars and all, okay. So, any battery that you can recharge, not use and throw, is based on this principle. Okay, this is sodium ion battery and this movement happens through an electrolyte solution. This movement happens through an electrolyte solution. Is it clear? Okay, so same way lithium ion batteries also work on the same principle. Okay, during charging positive to negative electrode, during discharging negative electrode to positive electrode. The only difference is here sodium ions are moving, in the lithium ion batteries, lithium ions will move. Okay, I hope you understood how this rechargeable batteries, sodium ion batteries or lithium ion batteries are working. Then you answer, which batteries are commonly being used now? Are we commonly using sodium ion batteries or lithium ion batteries? Very commonly we are using lithium ion batteries. Are very common. But we have a lot of problems with lithium ion batteries. Let us try to understand that. The problems associated with lithium ion batteries. First one, they are more prone to catching the fire or exploding. We keep hearing some electronic vehicle, a bike got exploded when it was on charging. We hear such news. Such incidents are more with lithium ion batteries. Comparatively, sodium ion batteries are safer. They do not explode like that. Okay, so that is one advantage of lithium ion batteries. Is it clear? Second advantage, they have longer lifespan. Okay, otherwise let us do it like this. Let us keep plus and minus. Okay, right. You tell me in the first one which is plus. Sodium ion plus, this is minus. Okay, second one, which have longer lifespan. Okay, lithium ion batteries have longer lifespan. So, they can be charged more number of times. Every night you are charging it. Your EV, you are charging it every night. So, like that many days you can charge it. So, this is plus, this is minus. Is it clear? So, that is a disadvantage of sodium ion batteries. Why lithium ion batteries are more commonly used than the sodium ion batteries? Because of this reason. And they have higher performance. So, this is also negative here. This is the reason why we are more commonly using, okay, lithium ion batteries because of their performance. Okay, next one is the lithium is a rare metal. Okay, these rare metals and minerals which can have a negative impact on the environment. Is it clear? Extracting the lithium, which is actually rare, will have a negative impact on the environment. So, this is minus here, this is plus here. It means sodium ion batteries are more environment friendly. Next one, they have higher energy density. What do you mean by energy density? Okay, small battery, more energy. Is it clear? Small surface area, okay, more energy. This is a huge plus point here and that is a minus point here, because let us suppose you are having a EV, okay, a small bike. In that you cannot keep such a huge battery. We need small battery, okay. So that is the reason why, okay, this is energy density, that is a plus point here. This is cheaper, plus point, negative point, okay. So I told you that in the lithium ion batteries, it is more energy density. It means small surface area battery can give more energy. That is the reason why this is suitable for portable devices like cell phone, okay, laptop, okay, EVs. For all these things, lithium ion batteries are in fact more suitable. But here, if you want to use a huge surface area, okay, like a different application where huge surface area battery is possible, in that case this is better. This is a total comparison of sodium ion battery and lithium ion battery. Now let us go back to the question after understanding it correctly. Is sodium ion battery cheaper? Yes. Not only cheaper, with lithium there is one more huge negative point. That is, okay, lithium is available only in 3 countries. Can you name the 3 countries? Argentina, Chile and Bolivia, right? Okay, so it means the total world need to export the lithium only from those 3 countries. We don't have much lithium resources. We have to import. Okay, let us import. But we don't have many sources to import also. We have to import from only 3 countries, which is called as lithium triangle. Okay, so which will also make it costly. That is another negative point. Okay, they are less likely to experience the thermal runaway. Yes, correct, safer. Environment friendly, correct, safer. But higher energy density, no. Okay, lithium ions have higher energy density. They can give more energy for small surface area. Is it clear? More energy per unit area. That is, in general density means what? Number per unit area. So they have more energy density. So second statement is wrong. 1, 3, 4 are correct. Okay. Now moving on to the next question. 11th question. Read the question. 40 seconds to go. Your time starts now. Okay, the right answer is D, as you guessed. Here I followed the regular pattern. In such questions mostly, okay, D is the right answer. Okay, yes, what 40% gave the right answer. Very good. First, okay. In fact, we already discussed about this. We know something about this gut microbiome. In one of the previous questions also we discussed, isn't it? Gut micro, in the topic called obelisk, in that we discussed. Yes, very good, CR7. Okay, gut microbiome. Now today let us know more about it. This is also called as normal flora. Okay, Chavi, Mukesh, Manish, very good, thank you. Okay, this is also called as normal flora. Okay, and what is this? This is friendly bacteria. This is friendly bacteria. Okay, so gut microbiome produce a variety of nutrients including short chain fatty acids, vitamin B and vitamin K. Did you notice whenever a doctor prescribes antibiotics to you, he will also prescribe a multivitamin tablet. Did you notice that? Whenever a doctor prescribes antibiotics, he will also prescribe multivitamin tablets. Did you notice? Why? What is the reason? Because your antibiotics like some amoxicillin etc., they do not know the difference between friendly bacteria and a pathogen. Is it clear? So, it will kill all bacteria. Friendly bacteria, pathogens, it cannot differentiate. It's not antibody, it is antibiotic. Biotic, it can kill the living organisms, the microbes. Okay, so that is the reason why our friends are also gone. Is it clear? So, two, when the friends are gone, okay, these friends are producing vitamin B and vitamin K for us. Now, nobody to produce vitamin B, vitamin K. So, we need to take the multivitamin tablets. Is it clear? So, that is the reason why you need to take vitamin tablets when you take antibiotics. Is it clear? So, this statement is correct. Next, okay. There are more bacterial cells in our body than the human cells, correct? Yes, correct. Interesting. You have roughly 3 times more bacteria. I mean, if you have 1 trillion cells of your own cells, you have 9 trillion bacterial cells. You have more bacterial cells in your body than your own cells. Is it clear? So, in a television conversation, I heard a sentence by somebody saying, I liked it so much. He was saying that, okay, it is difficult to say whether me, when I say I, when Bindu says I, who is this I? Is it that human being or is it that gut microbiome? Is it clear? Who is speaking with you, that gut microbiome or this person? Because I have more bacteria in my body than my own cells. So, who is actually me? Maybe me is that group of bacteria. It is that important it is. Is it clear? Gut microbiome controls metabolism. True. Did you hear some people, okay, not here, did you notice some people, how much ever they eat, they don't easily put on the weight. Some people, if they just smell the food also, they will put on the weight. What is the reason? The gut microbiome once again. The gut microbiome will help in the metabolism. Is it clear? So, some people have good metabolism, so they can easily digest even if they eat a little bit more. Okay, then, okay, so the gut microbiome controls our metabolism. You know something? It will ask you to eat one more chocolate. Is it clear? Okay, because like it wants food. So, it means when you are craving for a chocolate, you are not sure whether you are craving or that bacteria craving for the chocolate. Is it clear? Okay, gut microbiome controls our immune system. Yes. Okay, and it helps in digesting the cellulose. Yes, actually that is the reason why we are discussing it. This is the recent news from Hindu newspaper. Okay, so recently they discovered cellulose digesting. Okay, so whenever somebody scold you, why are you eating so much of sweet? Then tell them that no, it is not me, the bacteria are demanding that. Okay, coming back to this. So they recently identified a cellulose digesting gut microbiome. This is very important for us because, do you know, we have this organ called as appendix. Appendix produces an enzyme called cellulase, and it will digest cellulose. It will digest cellulose. Is it clear? Okay. We needed this when we were monkeys. But, okay, when the humans evolved, what happened? We became omnivores. We became omnivores. We started eating everything, not only the, okay, not only the, not only the plants. Plus we started eating the processed food. It means we peel off the skin, we mash it, we cook it, we boil it, we fry it. So where is cellulose? Cellulose already gone. Did you understand? It means we are not consuming much cellulose. So what happened? The appendix became vestigial organ. Vestigial organ. Vestigial means what? Inactive. Means it's not producing the cellulase enzyme anymore. It's not producing the cellulase enzyme anymore. Okay. So now, because of this, what happens? We do not have cellulase. Because appendix became inactive, we do not have cellulase. So what is the consequence of this? We cannot digest cellulose. Okay, what is the consequence of that? We need some bacteria that digest cellulose for us. Is it clear? Because you cannot digest cellulose. Your gut microbiome is digesting the cellulose for you. Okay, the total story. Appendix produce an enzyme called cellulase. It will digest cellulose. When we were monkeys, we had this organ functional. But when we evolved, we started eating the omnivorous, we became omnivorous and also started eating the processed food. So we are not actually consuming more cellulose. So we did not require this organ called as appendix. So the appendix became a vestigial organ. But we sometimes eat plant food also. Sometimes we also eat along with the peel. Okay. Then you need cellulase enzyme, but you don't have it. So this gut microbiome is digesting that cellulose for us. Is it clear? Okay. So that is the reason why, okay, this is also correct. And this is a new discovery. Is it clear? Okay. Right. Now that is all about the 11th question. All the above is the right answer. Yes. Now moving on to the 12th question. Read the question carefully. 40 seconds to go. Start. Unfortunately, I came from net only, Manish. Virus can be cultivated in the lab, but for that you need cell culture, Manohar. Right. Okay, the 12th question right answer is D. Only 2 are not true. Okay, so let us see what are they. Okay, which of the above, sorry, which of the above is not true? This is not true. Okay, right. Now let us start the discussion. Okay, Marburg virus. Who is Marburg virus? Ebola virus is there, na? Its cousin sister, related to the Ebola virus. Okay. Ebola virus and Marburg virus, they both belong to okay, the family called as filovirus or filoviridae. Filoviridae family or filovirus. Marburg virus, Ebola virus. They both are the members of filovirus family or filoviridae family. Is it clear? Plus, in that they belong to the genus called as the genus. Genus means what? Subgroup. For example, if this is family, one family will have many genus. Then what is a species? Another point of information. This is called a species. So species means, okay, a smaller group. Species is a smaller group, genus is a bigger group, and family is a much bigger group. Is it clear? Yes. So, Marburg virus and Ebola virus, they both belong to the family called as Filoviridae, and they both belong to the genus called Nipah virus. They both belong to the genus family Filoviridae. Family Filoviridae and genus Nipah virus. Okay, and Rosetus aegypticus fruit bat is the natural host of the Marburg virus. Another information is, okay, Rosetus aegypticus fruit bat is a natural host of the, okay, Marburg virus. Is it clear? Marburg virus is transmitted to people from the fruit bat. How? For example, we eat fruit, okay, without properly washing it, but it could have been, okay, already eaten by or touched by this bat. Is it clear? Then we will get that particular virus from the bat by consuming these fruits. Is it clear? Okay. Then, so this is Rosetus aegyptiacus, right? Okay. Marburg virus disease causes severe viral hemorrhage, just like Ebola virus. It will cause severe, okay, viral hemorrhage fever in the human beings. And the fatality rate means what? Okay, like how many people will die if 100 people will get infected. That ranges from 24 to 88% with an average of approximately 50%. It means roughly 50% people will die. If 100 people will get infected, 50 people will die. But it depends on the availability of the treatment. For example, if you are living in a developed country, advanced country with full medical infrastructure availability, then your chance of death can be only 24%. But if you belong to some sub-Saharan country where these viruses are more common in Africa, then the chance of death is more. So average is 50%. Early supportive care with rehydration, symptomatic treatment improves the symptoms. And we do not have a special medicine for Marburg virus. Is it clear? No special medicine for Marburg virus, but symptomatic treatment. I mean, if you get fever, fever medicine. If you get body pains medicine. If you get dehydrated, rehydration, like that. So that is how we have to protect the people. This is about the Marburg virus. Is it clear? Then let us now see about the Ebola virus, its cousin sister. Okay. So Ebola virus is an RNA virus. Okay. And what do you mean by this? See, a virus will have some more basics here. A virus will have either RNA or DNA, never both. Either RNA or DNA plus a protein coat. Either RNA or DNA plus a protein coat. So viruses are two types, RNA viruses, DNA viruses. RNA viruses are again two types, okay, retrovirus, RNA virus. RNA viruses retain RNA, but retroviruses, the RNA will get converted to DNA. The RNA will get converted to DNA, that is retrovirus. Retrovirus example HIV and HTLV. Another virus called HTLV. RNA viruses, most of the animal viruses are RNA viruses. Zika, Nipah, Ebola, Marburg, okay, coronavirus, flu virus, hepatitis, all these are RNA viruses. Is it clear? Okay, and DNA viruses are rare, very rare DNA viruses we have. Okay, and Ebola is not a DNA virus, it is not a retrovirus, it is an RNA virus. Okay, now let us deal with this question. Marburg virus belongs to the same family, Filoviridae, as Ebola, correct. Okay, Ebola virus disease cause bleeding, hemorrhage. Okay, because it damages the blood platelets. Blood platelets are also called as thrombocytes, and they help in blood clotting. They help in blood clotting. Is it clear? Okay, and these Ebola virus, Marburg virus, both, they damage this blood platelets. So that is the reason why it will lead to blood, like bleeding, hemorrhage, internal bleeding and all that. So this is correct, this is correct. Only this is wrong because it is an RNA virus but not a retrovirus. Okay, is the 12th question clear? Which is more fatal, Ebola, Marburg? Both are cousin sisters, both fatality same, 24 to 80%, average 50 to 60%. It all depends on, okay, many factors, availability of medicine, medical infrastructure, okay, plus sometimes, for example, when you get COVID, okay, when there is no wave, then you may survive because you will get hospital bed. Medical infrastructure is not overloaded. In the first wave, second wave, many people died, okay, not because they got severe disease, but because they did not get the treatment at the right time, because the medical system is overloaded. So, if you get Marburg virus when there is no outbreak and you get Ebola when there is outbreak, then the Ebola will have more fatality. So, these are the multiple factors we have. Right, that is all about the 12th question. Moving on to the next question. This is about influenza. Very important. Did you notice last 6 months at least 6 to 7 articles were there? Okay, 6 to 7 means not all, only Hindu 6 to 7 times given about influenza. Do you agree? Okay, repeatedly there are articles about influenza, bird flu. Okay, so let us suppose, so I decided to discuss about flu. Okay, I just use this question as an opportunity, but I will discuss everything about flu. Let us know everything about flu. Okay, consider the following questions. Okay, consider, 40 seconds to consider. Done already, super fast Mukesh. Okay. Now, the right answer for the question is, okay, A, only one. How many are true? Only one. Which is correct? This is correct. Statement number 1 is correct. Okay, right. All influenza viruses are RNA viruses. I just told you. Not only influenza, most of the animal viruses are RNA viruses. Okay, you just have to remember 1 or 2 retroviruses, 1 or 2 DNA viruses. Remaining all are, don't by heart RNA viruses. That's a huge list. By heart 1 or 2, HIV, HTLV are the retroviruses. 1 or 2 DNA viruses you have. By heart them. Okay, that's it. Remaining all are RNA viruses, and influenza viruses are all RNA viruses. That is correct. Bird flu, H5, this is about this only you have these many articles. Every newspaper in the last 6 months published at least 6 to 7 articles on flu, bird flu. That is about H5N1 only. Okay, it is an epizootic enzootic disease. Greek and Latin, isn't it? What do you mean by this? Okay. Do you know these words epidemic? Demic is about human population. You know demography, is it clear? Okay, this is about human population. Okay, so replace demic with animal, because this is bird flu, na? Birds are getting infected. Then the word is epizootic. I hope you understood this. Okay, then same way, enzootic. Did you hear about endemic diseases? Like for example, malaria in Africa and some parts of India. Endemic. Endemic means what? Confined to one region. So replace demic with zootic, enzootic. Okay. You know pandemic? Everybody knows about pandemic after COVID-19. Who does not know? Okay. So in the pandemic, okay, replace the word demic with zootic, panzootic. So panzootic means all the world. Okay, all the world. Okay. Enzootic means one region, confined to one region. Is it clear? Okay. But always, no season, always this is a problem, like that. Whereas epidemic or epizootic means outbreak. It means seasonal. Short duration, many cases will come. That is called as epidemic. Is it clear? Okay. So, is this epizootic, enzootic? No, this is panzootic. Now you have this bird flu all over the world. So, this is a wrong statement. Is it clear? I could have given only epizootic, right? I have given this word also only to tell you these three. Epizootic, enzootic, panzootic words introduce. Okay, is my target. So, that is the reason why this statement is made like that. Okay, next. Avian influenza belongs to influenza type D. No. It belongs to influenza type A. Okay, it belongs to influenza type A. Okay, so how many of the above statements are true? Okay, only one statement is true, that is they are all RNA viruses. Okay, question is gone. But let us know more about influenza. Okay, so that's why don't count the number of questions, because I am not targeting to give that fishing man in between like questions. I can make 50 also, but that is not my target. My target is to give you as much information as possible and a few questions to target. Is it clear? Practice. Okay. Now let us see more about influenza. Avian influenza or bird flu refers to the disease caused by the infection of avian influenza type A virus. Bird flu is type A virus. Frequently, the virus can infect mammals also. Okay, as of now, mink, ferret, seal, cat, cattle, and recently, okay, one human being working with the cattle also. Okay, a herdsman also got this bird flu. Is it clear? Okay, so all these mammals are also getting the bird flu. Is it clear? Okay. So, we have all the mammals are also getting the bird flu. Now, symptoms in the human beings. If a human gets bird flu, how does it look like? Mild to severe. It all depends on your immunity, your age, and your genetics, and your food habits, the other diseases you have. So like this, whether you will get mild symptoms or severe symptoms depends on all these things. And the symptoms will begin like fever, cough, sore throat, muscle pain, nausea means vomiting sensation, abdominal pain, diarrhea means loose motions, vomiting, etc. People can also develop severe respiratory illness, like the people died during the COVID pandemic second wave, like that. With respiratory failure, even the death, acute respiratory distress, viral pneumonia. What is pneumonia? Fluid accumulation in the lungs. Fluid accumulation in the lungs. Okay. Types of influenza virus. Symptoms over. Next one, types of influenza virus. I told you that bird flu belongs to influenza type A. Okay, that is type A. Then what are the other types? Okay, we have 4 types of influenza. A, B, C, D. Is it clear? 4 types of influenza. Okay, influenza A, okay, is birds, mammals, humans. Is it clear? Influenza A infects birds, mammals and human beings. Correct? Next. Influenza B, okay, humans, pigs and seals. Humans, pigs, like swine flu. Pig swine flu, like that. Humans, pigs and seals. Influenza C, mainly in the human beings. Seal is a sea mammal. It lives in the ocean. Okay, right. Mainly occurs in the human beings, but found in the dogs and pigs also. Okay, influenza D mainly affect the cattle. Bovine flu we call it as. Bovine flu. It mainly affects the cattle. Okay, that is cows, buffaloes, all these things. Is it clear? Okay, it is not known to infect the human beings yet. So, what is the only type of influenza that is still not known to infect the human beings? Influenza D. The influenza that is not known to infect the human beings is influenza D. Okay. Avian influenza, aves means birds. So, bird flu is type A virus. Type A viruses are classified based. Okay. Now, what we are discussing is C. Influenza A, B, C, D. Now what we are discussing is subtypes of A, B, C, D. Is it clear? Now what we are discussing is subtypes of A, B, C, D. Okay. We are now classifying A based on 2 proteins. Okay. The first one is called as heme-agglutinin. Try to observe the word. Heme means blood. Agglutinin. Heme-agglutinin. Okay. And next one is neuraminidase. Neuraminidase. Heme-agglutinin is H. Neuraminidase is N. Clear for all of you? Okay. Heme-agglutinin is H, neuraminidase is N. These are the two proteins in type A virus. Is it clear? 2 proteins. Now, okay, this classification is based on, okay, types of H and N. Did you understand the class? H1N1, H5N1. What is that? For example, okay, H5N1 means fifth type of heme-agglutinin, first type of neuraminidase. I hope you got the point. H1N1 means first type of heme-agglutinin, first type of neuraminidase. Is it clear? So, influenza A, B, C, D not classified. Only type A is classified into. Okay, remember this. B, C, D are not further subclassified. only type A is subclassified into these types. Is it clear? So, these are all. Avian is H5N. So, should you remember all these things? No, don't worry. Remember this one. Whatever is more in the news, this is more important. That's it. Others you don't have to remember. H1N1 is important. Okay. H3N2 is important. That's it. Because why these are important? Because they are more common in the human beings. Why this is more important? Because it is a panzootic and it is also infecting all the mammals. So there is a risk that it can cause a pandemic also. Presently it's a panzootic, but in future it can cause a pandemic also. So that is the reason why these are important. So don't worry, should you remember all these things? No need of remembering all these things. What all you have to remember is these two, because they are common in the human beings. H1N1 swine flu, okay. H5N1, okay, bird flu. H3N2 is also important. That's it. These are the things you have to remember. Okay. Now, this is the story of influenza viruses and the answer is already there. Now, moving on to the 14th question. This is about hepatitis. Okay. Read the question. 40 seconds to go. Your time starts now. Not the last question, this is marathon. Oh. Nafisha, I told you it is suspense. Actually, I was about to say and I stopped. I don't want to tell you total how many questions. Okay. 14th question, the right answer is C, 1 and 3. Okay. So now, leave the question. Let us know more about hepatitis. Okay. Hepatitis is an inflammation of liver. The word hepatic means liver. The word itis means inflammation. Okay. So you can add, see the biological terms are easy to understand, remember also, because there will be some common root words. Itis means anything like laryngitis, pharyngitis, meningitis, hepatitis. So like that, itis is a common root word here. Okay. Now, hepatitis is an inflammation of liver that can be caused by viruses or even non-infectious. I mean, alcoholism also can cause hepatitis. Multiple reasons are there. Accumulation of toxins, okay, like that. There are total 5 types of hepatitis. Just like we have seen 4 types of influenza virus, there are 5 types of hepatitis virus. Is it clear? Each type cause liver disease only, but they vary in intensity. They vary in the intensity and also mode of transmission. Intensity and mode of transmission. Is it clear? Okay. So, that is about the types of virus. Is it clear? Hepatitis B and C are the most common causes of liver cirrhosis. What is liver cirrhosis? Memorize the word please. Liver cirrhosis means degradation of liver cells. Degradation of liver cells. Is it clear? Liver cancer and viral hepatitis related deaths. It means total A, B, C, D, E 5 types are there, but B, C You can remember it like BC category, reservation BC category. B, C are the most dangerous ones. They cause liver cirrhosis, liver cancer and death due to severe hepatitis. That is about BC. Okay, right. Then, now let us see the types of hepatitis and the mode of transmission. How do we get that? Hepatitis A, it is water contamination and feco-oral route. Okay, defecation and that entering into the water, contaminating the water and consuming that water. That is the reason. Hepatitis B, contact with HIV in body fluids like blood, vaginal secretions, semen. Means this is a sexually transmitted disease. Is it clear? That is a sexually transmitted disease. Next, C. This is also same, body fluids, blood, vaginal secretions, semen, but more through blood. Okay, more through blood and rarely through sex. Rarely through sex. Okay, remember that. Hepatitis D, contact with blood containing HDV. This is also mostly through blood. Okay, so E, exposure to food and water. Remember A, E, first one, last one, food and water. In between three are body fluids. Is it clear? First one, last one, food and water. In between three are body fluids. In that, the exactly, okay, say, first one, okay, that is mainly through sex. The other two, these two are mainly through blood and also through sex. Is it clear? Easy to remember? Okay, next. Coming about vaccines. We have a vaccine for A and we have a vaccine for B also. CDC recommended hepatitis B vaccine for all the newborns. There is a series of 3 doses given in the first 6 months of the childhood. Is it clear? In India currently, okay, children below 6 months age are getting 3 doses of this hepatitis B vaccine. Okay, in India initially hepatitis B vaccine was rolled out, okay, in some cities and districts in 2002 and 3. By 2011 and 2012 the vaccine is available for all the country and also it became a part in the integrated child immunization program. Like Indradhanush, in the government program will be there, right? Okay, vaccination, child immunization program, it became a part of that. Is it clear? Expanding its reach and effectiveness in preventing the disease. Is it clear? And remember, these vaccines, the more people take the vaccine, the more we get protection. It means, if you take vaccine, I will get protected, because the unvaccinated people are like the fuel for the fire. The unvaccinated people are like the fuel for the fire. So they are the source where the virus will survive and that virus can infect the other people. Immunized people also can get it sometimes. But if more people will get vaccinated, that fuel is completed. So the fire will actually stop. Okay, so that is the reason why I will not get protected only if I take the vaccine. I will get protected only when everybody will take the vaccine. Is it clear? Okay, vaccination against hepatitis B can also prevent hepatitis D, but there is no vaccine for it. Means for on the whole, okay, C, D, E, no vaccine. Is it clear? C, D, E, no vaccine. You can remember like this. Last 3, no vaccine. First 2, there is vaccine. Same way in the A, B, C, D, E. These 2 are water. These 3 are body fluids. Then in this also, okay, this is mainly through sex. These 2 are mainly through blood. I hope you can easily remember this visually. Right. So this is all about, okay, the hepatitis virus. Now let us check the question. Okay. There are currently no vaccines for CDE, correct. Okay. HCV is 50 to 100 times. HCV means what? Hepatitis C, okay, don't get confused. Hepatitis C is also called HCV. HCV means hepatitis C virus. Is it clear? HCV means hepatitis C virus. Okay. No, wrong. If you remember, actually it will mainly spread through, yes, fill my sentence. It will mainly spread through, very good, Varsha. Okay, through blood, right, rarely through sex. So it is not 50 to 100 times more sexually transmitted than HIV, no. B and C are the most common cause of liver cirrhosis, correct. Okay, so 1 and 3 are true with respect to hepatitis. Is it clear? Okay, next. Hepatitis completed. Moving on to the next question. Okay, consider the following statements. Come, quickly consider. 40 seconds to go. Your time starts now. Okay, 15th question, the correct answer is B. B is the correct answer. Okay. 40% gave the right answer, good, going good. First statement, I already told you, do you remember? In this class only I told you. Okay, University of Oxford produced a vaccine for Nipah virus using ChAd-OX1. Okay, chimpanzee, adenovirus. Okay, this is serial number. Okay, ChAd-OX1, imagine this is ChAd-OX1. This is our Covishield. Did you recognize? This is our Covishield, the vaccine that most of us have taken. Okay. Then okay, so like this, carrier is same. So if you keep SARS-CoV-2 gene in that, it will become SARS-CoV-2 vaccine. If you keep Nipah virus gene in that, it will become Nipah virus vaccine. If you keep Ebola virus gene in that, it will become Ebola virus vaccine. I hope you understood the concept. So Oxford University is developing ChAd-OX1 based viral vector vaccines. All these are viral vector vaccines. Okay, for all these diseases. So correct. Fruit bat Pteropodidae family are the natural host of the Nipah virus, correct. Okay, we have seen this, no, actually we have seen this about Marburg virus, but the same fruit bat is the natural host of Nipah virus also. Is it clear? This Pteropodidae family fruit bat, okay, is the natural host of Nipah virus and Marburg virus. Okay, right. Hendra and Nipah, just like Marburg and Ebola were cousin sisters, Hendra Nipah virus belong to same Nipah virus category. Is it clear? Recently developed monoclonal antibody targets the attachment protein G of the virus. This is a wrong statement. Okay, this is the wrong statement. So, this is about the news. What is there in the news recently? Okay, so let us try to understand that. Antibody therapy for Nipah. Antibody therapy for Nipah. Nipah virus is a paramyxovirus that causes encephalitis in the human beings. Encephalitis means what? Okay, encephalon is a layer that covers the brain. So, this is a kind of a brain infection, we can say. Okay, to envelope glycoproteins. Envelope glycoproteins means what? I told you that a virus will have RNA plus a protein coat, I told you, isn't it? That is nothing but envelope glycoprotein. That is nothing but envelope glycoprotein. Okay. So, this envelope glycoproteins are two types. First one is attachment protein. First one is attachment protein. Okay. And one small correction. I think I told about this Nipah virus for that Marburg. Please cross check once. Okay, somewhere it got jumbled. Hendra and Nipah virus belong to Nipah virus. Make that correction. This is not regarding the Marburg virus. Hendra and Nipah belong to the Nipah virus category. Is it clear? Hendra and Nipah virus belong to Nipah virus category. Make that correction. Okay. Right. Now proceeding forward. Right. So this will have two types of things. Okay. That is attachment protein. Okay. Called as G protein. Second one is called as fusion protein, called as F protein. Second one is called fusion protein, called F protein. Okay, they both just like in the flu you have this H1, H and N, hemagglutinin and neuraminidase. Same way here you have G protein and F protein. Is it clear? In the flu virus we have hemagglutinin neuraminidase. Same way here you have G protein and F protein. Where are they? In the envelope, in the coat of this virus. Okay. Which virus? Nipah virus. Is it clear? Okay. Nipah virus. This is RNA. This is the protein coat. I repeat, Nipah virus. This is RNA. This is the protein coat. The coat is nothing but the envelope glycoprotein. It has two proteins in that. First one is attachment protein called G protein. Second one is fusion protein called F protein. So recently the news here is they developed an antibody. They developed an antibody for F protein. Is it clear? Okay. A monoclonal antibody known as, you need not remember the name, don't worry. Even this FG also I don't think you have to remember. See, how to use the news is very important. Let us suppose what is there in the news. Listen this very carefully please. What is there in the news? In the news you have this one HU1F5. Okay. Same way you have G or RBP and F. These are there in the news. But they will not ask question on this. They may ask something about Nipah virus. They may ask something about Hendra virus. Is it clear? Okay, I don't want to keep you in the suspense. I'm planning to discuss up to 20 questions. Okay, let us see how it goes. Okay, and I will take at least till 10:15, or I may try to extend till 10:30. That is the time and the number of questions details. Okay, I don't want to keep you in the suspense anymore. Not more than 21, 22. Okay, right. So now coming back to this. Okay, now I hope you understood how to use the news. Okay, it does not mean that all these scientific terms they will ask. No, they may ask something about these viruses. Is it clear? Something else about the virus, like that. Okay, I don't expect that. I don't think that you have to remember these names, but just try to understand. So here they made an antibody for the fusion protein. This is a drug. Tell me, what is the drug here? What is the medicine here? The antibody for the fusion protein. The fusion protein will get disintegrated. Then the Nipah virus will die. That is a treatment. Is it clear? Okay. So the new recently developed monoclonal antibody targets the attachment protein wrong. It will target the fusion protein. Is it clear? Done? Thank you so much, Manish. Moving on to the 16th question. Okay. Read the question carefully. Your time starts now. Okay, the correct answer is B. Only 2 are true. Which 2 are true? 1 and 3 are true. This was very much in the news. I hope you remember. Recently, everybody was afraid of this also. AstraZeneca in the court admitted that some of the people who have taken their vaccination are getting this blood clotting problem. Okay. So, everybody who took this Covishield got afraid also. This question is regarding that. Okay. Right. Thrombotic thrombocytopenia. This is a technical name. Basically, what is this? Blood clotting. Okay. Okay. I'll try to elaborate. Thrombosis means blood clotting. Okay. Then thrombocytopenia is like okay, decreased blood platelets. In the blood. Decreased blood platelets in the blood. Okay, right. And they both are interconnected. For example, if there is thrombosis, do you know what causes blood clotting? Thrombocytes cause blood clotting, isn't it? Thrombocytes cause blood clotting. Isn't it? So, now let us suppose this is the blood vessel and all the thrombocytes, that is blood platelets, are here. Did you get my point? These are all the thrombocytes. So, if all the thrombocytes assemble here In general, in the blood, what happens to the number of thrombocytes? You tell me. The number of thrombocytes in the blood, beautiful, decreases. Very good, decreases. Why? Because whatever thrombocytes you have, they all got stuck here. So you don't have the thrombocytes in other parts of the blood. Is it clear? So, okay, that decreased platelet count is called thrombocytopenia. This is called thrombocytopenia. This is called as thrombosis. Is that clear? The clot is thrombosis, and the decreased count of thrombocytes or the platelets in the blood in general is called as thrombocytopenia. Both will occur together. It means the thrombocytopenia is because of the thrombosis. Do you agree? Thrombosis causes thrombocytopenia. The blood clotting here is causing the decreased blood platelets in the blood in general. Is it clear? So, therefore, we call it as, okay, thrombocyte is equal to platelets. Okay, deductivist, thrombocytes is equal to blood platelets. Yes, both are the same and they cause blood clotting. Right? Okay. So, that is a story of thrombotic thrombocytopenia. If you observe, I used this word thrombotic thrombocytopenia 5-6 times. Why? With the hope that you will memorize this word. Try to memorize the word. Thrombotic thrombocytopenia is a rare side effect of the viral vector vaccine made from ChAdOx1 platform. What is that? Covishield. Okay. The vaccine made on ChAdOx1 platform for coronavirus is Covishield. Yes. The vaccine increases VLDL, very low density lipoproteins. No, that's an irrelevant statement. I just kept it. It has nothing to do with very low density lipoproteins. Okay. Then the vaccine causes immune system to make antibodies. Okay. To activate the platelets and cause the blood clot. Okay. This point is explaining the mechanism. This point is explaining the mechanism. Okay. So, what is the mechanism? How it causes? Okay. So, when you take Covishield, I will explain it. Okay. You have taken, remember it like a flow chart. You have taken Covishield. Okay. Then what happened? It produced antibodies. Then what happens? These antibodies bind to platelets. These antibodies bind to platelets like this. Okay, this is platelet. Okay, platelet means thrombocyte. Okay. Then what happens? This thrombocyte gets activated. The thrombocyte gets activated. Okay, right. The activated thrombocyte will cause clotting. That is the mechanism. Okay, I repeat the mechanism. You have taken Covishield. It produced the antibodies. Which antibodies? Okay, antibodies for SARS-CoV-2 are produced. These antibodies will bind to, look at this, these antibodies will bind to, okay, blood platelets, and the blood platelets will get activated. The activated blood platelets will cause blood clotting. The blood clotting also causes decreased platelet count. Is it clear? That's it. That is thrombotic thrombocytopenia. Thrombotic thrombocytopenia. Is it clear? Okay. So, the same mechanism is discussed here. The vaccine causes immune system to make antibodies that will activate the platelets and then cause the blood clot and subsequent drop in the blood platelets. Can you correlate this sentence with that? See, this only one sentence is equal to this complete flow chart. Is it clear? Vaccine causes immune system to make antibodies that will activate the platelets and cause the blood clot and subsequent drop in the blood platelets. So, this is correct. 2 is the wrong statement. Okay. Then, one point is there is a blood thinner called as heparin. It is actually a blood thinner, but this will also cause similar side effect as. This will also cause similar side effect as Covishield. Okay. Similar to the side effects of Covishield. Done? Clear for all of you? There is a medicine called heparin. Why do we take heparin? It is a blood thinner. Some of your families, maybe some grandmother, grandfather, if they have cholesterol, okay, there are some situations where the blood thickness increases, then they may take this heparin injection. Okay, heparin is a blood thinner, but it is also having the side effect, same side effect. What is that side effect? Thrombotic thrombocytopenia. Yes, clear for all of you. So, that's all about the question number 16. Moving on to the next question. The question is right in front of you. Read the statements carefully. Participate in the poll. Your time starts now. Yes, Manshi, very good question. Your question is indicating that you are understanding the concept correctly. That is a blood thinner, but it causes blood clotting. That is side effect. Intended effect is blood thinner. It is expected to thin the blood, but its side effect is blood clotting. Rarely it will occur. Manshi, I hope it is clear. Yes. Now, so the right answer for 17th question is D. Okay. Simple question. Which of the above are not true? None of the above. It means all are true. Okay. Right. Now, liquid nitrogen is used for preserving the food. Okay. First of all, what is this liquid nitrogen? Nitrogen is N2 and at room temperature, it is in the form of gas. At room temperature, it is in the form of gas. So, liquid nitrogen is minus 196 degrees Celsius. Minus 196 degrees Celsius. At such a low temperature, if you keep something, it can be stored up to 100 years also. Okay. So, we can use it as a preservative for food, and we can use it in the cancer treatment. Then you may wonder, how can we treat cancer using this? Is it clear? So, here what they do is, the cancer tissue is frozen. Okay. The cancer tissue is frozen. Is it clear? Okay, and see, this is something like these days they are doing this for fat reduction also. What they do, they freeze that fatty layer and then after some time that frozen thing will be disintegrated. Is it clear? So that is how they are doing. Here what they do, they freeze the cancer tissue. Okay, and later this frozen part will be disintegrated. The body will damage it later. Okay, so that is how liquid nitrogen can also be used in the cancer treatment. Is it clear? Next, we can also use it in oocyte. This is about egg bank. Egg bank. Okay, and sperm. This is about sperm bank. And stem cell. This is about stem cell bank. And okay, somatic cells, embryonic cells. Okay, all these things can be frozen. But why do you freeze? For example, let us suppose you are now 23-year-old, but you have a plan to marry at 40. But you heard that, okay, like genetic diseases increases. Genetic diseases increases as the age increases. Is it clear? Okay, then what you can do is when you are 23, you can collect your egg cells, store it in the egg bank and you can have a healthy baby when you are 40. Is it clear? So that is also possible. And sperm bank. Is it clear? Sperm cells can be frozen and donated to the people who want to have the children, but the man is unable to release the sperm cells due to some health condition. So same way stem cells can be preserved to manufacture organs from that. To manufacture organs from that. Is it clear? Somatic cells also can be frozen for the future use. Okay, for cloning purpose. For cloning purpose. Embryonic cells, same thing. Let us suppose you are 23 but you want to have the children at 40. So you can take your egg cell, fuse it with the sperm cell, form the embryo and then freeze the embryo. After 20 years, you can transfer that embryo into the womb and can have the baby. So all these things you can do with the liquid nitrogen. Uniform cooling of the concrete mixture, building construction, concrete mixture. Okay, the concrete mixture should immediately cool down. Otherwise, before it cools, if you get rain and all, it is very risky, isn't it? For example, you have put the slab. Okay, and if the concrete mixture is not quickly getting cold, it will remain in the liquid condition. In that time, if the rain comes, then the total building is at danger. So that is the reason why to quickly cool down this concrete mixture, that to uniform cooling, okay, we can mix liquid nitrogen in the concrete mixture. Is it clear? Yes, these are all the applications of the liquid nitrogen. Okay, why this is in the news? Recently, a small boy died unfortunately by eating a smoky biscuit. Did you hear about it? A smoky biscuit he consumed and he died because that is full of liquid nitrogen. It perforated the digestive system and that poor boy died. In that context, it was in the news. Okay, and also like we should not use it. Okay, in that context, the rules also became strict that we should not use, okay, liquid nitrogen in the food items. They must be used only for preservation, not for consumption. Liquid nitrogen should not be used for the consumption. Okay, so that is the point. That is about the liquid nitrogen. Okay, question number 18. Read the question carefully. Your time starts now. Yes, they are used in ice creams also, but we should not consume such things. Right. 18th answer is A. 1, 2 and 4. Okay. Now let us see about Voyagers. Voyager 1 and Voyager 2, they are the two spacecrafts launched in the year 1977. Hopefully, they will celebrate their 50th birthday also. This is the longest duration stayed spacecrafts. These are the first spacecrafts to cross the heliopause. Heliopause. What is heliopause? Okay, this is the border between solar wind and interstellar wind. Solar wind and other stars wind. You know what is solar wind? Is it clear? That particles one. Okay, right. Sun is a star. Same way other stars also will have their own particles getting released. So the border line between solar wind and the other stars wind is called as heliopause. If you cross heliopause, you will go into interstellar space. If you cross heliopause, you will go into interstellar space. Voyagers are the first spacecrafts in the world to cross the heliopause and enter into the interstellar space. Is it clear? Okay. See, Voyager 1 was originally sent to study Jupiter and Saturn. Is it clear? It means it did more action, more activity than it is expected. Is it clear? Far beyond our expectations. We after all wanted it to study about Jupiter and Saturn. Okay, but it actually studied all these things, crossed the sun and entered into the interstellar space also. Voyager 2 went to explore the Uranus and Neptune. Same way, it also did more than expected. Okay, we after all sent it to study, go study Uranus, Neptune and come back. Then it said, no, I will not come back, I will just keep on going. So that is Voyager 2. And Voyager 1 entered into the interstellar space in the year 2012. And Voyager 2 entered into the interstellar space in the year 2018. Is it clear? Then, now comes to the question, what is the fuel used? Okay, so that it sustained the spacecrafts for approximately 50 years. What could be the fuel? The only possible fuel that can sustain it for 50 years is, okay, radioactive fuel. Is it clear? Okay, nuclear fuel. Because you may think that it is solar, but not possible. Think about Chandrayaan 3's rover. Chandrayaan 3's rover. 14 days gone. Why? Because moon's day completed, moon's night started, gone, it slept. Is it clear? So that is the reason why you cannot depend on, okay, you cannot depend on solar fuel. This liquid, solid, cryogenic fuels, how much fuel you have to fill so that it will keep going for 50 years? How can you fill that much fuel? So solid, liquid, cryogenic fuels will not work. Solar will not work. The only thing is nuclear. What they do is they use the radioisotope. They use radioisotope, thermoelectric, generator. Okay. So in this there are two units. First unit is called as heater unit. heater unit. In the heater unit, what happens is plutonium, Manisha, thank you, Manas, thank you. We use plutonium, okay, will undergo radioactive decay. When it undergoes radioactive decay, it will generate heat. It will generate heat. This is what happens in the heater unit. Okay? Yes. I repeat, Voyagers contain radioisotope thermoelectric generator RTG. This RTG is having two components. The first component will have plutonium isotope in that, okay, and it will undergo radioactive decay. This decay releases heat energy. So therefore it is called heater unit. The second component is called as generator unit. Generator unit. It will convert heat to electricity. It will convert heat to electricity. Therefore, it is called as, look at here, that is the reason why it is called thermoelectric. Okay, why? Heat to electricity. That is the reason why it is called thermoelectric generator. This is the reason why Voyagers are able to run for such a long duration. Is it clear for all of you? Yes, that is about RTG story. Okay, and now comes the question, then why don't we use it in the other spacecrafts? Yes, good idea, we are using it. Voyagers are not the only spacecrafts where RTG is used. This is used in, of course, Voyager, Cassini, Curiosity are the other spacecrafts that used RTGs. Is it clear? Okay. Plus, ISRO and BARC together is also working on, okay, using the RTG for its, okay, spacecrafts. I repeat, RTG is not used only in the Voyagers. NASA already used it for the other spacecrafts like Cassini and Curiosity. Plus ISRO is also trying to, it is working on the RTG component for its future spacecrafts. Is it clear? Okay, right. So, radio thermoelectric generators, right, BARC and ISRO, yes, that point is also given here. I hope all the points covered, isn't it? Okay. Now, yes, plutonium half-life is 88 years. Okay, plutonium half-life. What do you mean by half-life? Okay, half-life of plutonium, half-life of plutonium is 88 years, means it will take 88 years for half of the plutonium to undergo decay. Is it clear? That is the meaning of half-life. Then you may ask me, why 50 years? Why can't Voyagers celebrate century? Okay, yesterday at least they could not hit the century, no one. So at least why can't Voyagers hit the century? Okay, because yes, 88 years, half of it is completed. Now half of the fuel is not completed. Okay, but if little fuel reduces also, they do not have enough fuel to run the space. They have the fuel. Plutonium is not exhausted. Okay, they have the fuel, but they don't have enough fuel to run the Voyagers. Is it clear? Okay, so that is the reason why they are about to complete their life. So what NASA is doing is, for example, sometimes in our mobile phone when we have very little charge, only 10% or something, what we do? We keep it in the energy saving mode. Whatever apps we don't want, we try to kind of switch it off. Same thing NASA is doing. In the Voyagers, whatever components are not crucial, NASA is switching off all of them. So presently Voyagers are in the energy saving mode. So like sentimentally, emotionally, NASA wants to celebrate the 50th birthday. Decided that I will celebrate 50th birthday of Voyagers. So this is the story of Voyagers, right? Okay. Now, let us see this question. Voyagers use plutonium, correct. Voyager 2 is the only spacecraft that studied the 4 giant planets of the solar system, Neptune, Uranus, Saturn, Jupiter, from the close range. Yes, it's a fact, remember. Voyagers are the only spacecrafts to use the RTG? Wrong. Okay, so like Cassini, Curiosity also used it, and ISRO Bark are also trying to develop an RTG component for us. They are the first spacecrafts to cross the heliopause, correct. Okay, right, that is the 18th question. Now moving on to the 19th question. Okay, right. Consider the following statements. Very interesting question. Okay, 40 seconds to go. Your time starts now. Okay. 19th question, the right answer is C. All the 3 are incorrect. All the 3 are incorrect. Okay. Fine. Now let us try to understand the concepts given in the statements. First, okay, what is Darwinism? You all know what is Darwinism? Survival of the fittest. See, there is a very important life lesson here in this Darwinism, that is, there is a beautiful quotation. It is not the survival of the strongest. It is the survival of the fittest. I mean, if you can exactly fit in the given situations, then you will become successful in the life. Okay, so that is not strongest. A strongest tree can collapse when the rain comes. A small plant which is very flexible and can easily adjust, that will survive. It is not the strongest, but it is the fittest that will survive. Now coming back to this, okay, natural selection, nature will select the fittest one. So this is about Darwinism. But Darwin did not know genetics. Okay, it means Darwin spoke about variations, and the best variation being selected, he spoke. But okay, he did not understand what causes variations. Okay, Neo-Darwinism clubs Darwinian evolution with Mendelian genetics. Okay, who is Mendel? Okay, father of genetics. Mendel is father of genetics. Is it clear? Okay, so it means mutations cause variations. Mutations cause variations, and the fittest variations will be selected. Fittest variations will be selected by the nature. This is called as Neo-Darwinism. Is it clear till now? This is called Neo-Darwinism. Okay. Then there is one more theory called endosymbiotic theory. Okay. Let us try to understand this endosymbiotic theory. Okay. Very interesting. Listen carefully. There were 2 bacteria. Okay. This is bacteria A. Okay. This is another bacteria, a bacteria B. Okay. Now, it entered inside the bacteria A. It means A eat away B. Did you understand? And then, okay, A will give shelter to B. A will give shelter to B and B will perform respiration for A. B will perform respiration for A. Is the story clear? There were two bacteria. Okay. A eat away the B. Then A will give shelter to B. And B is performing respiration for A. Okay. So, can we call this relation as symbiosis? You all know the meaning of the word symbiosis. Symbiosis means mutual help. Symbiosis means mutual help. Right? Okay. Plus, we are calling it as endosymbiosis. Endo means what? Inside. One is inside the other and they both are symbiotic. Is it clear? One is B is inside the A and they both are symbiotic. Endosymbiosis. Right? And you know what is B? Our mitochondria. Did you guess this? Okay, the organelle that performs respiration, the powerhouse of the cell. This is how, okay, A became a eukaryotic cell. I hope you know prokaryotic cell and eukaryotic cell. Prokaryotic cells do not have organelles, but eukaryotic cells have organelles. Mitochondria is an organelle. Okay, when A eat away the B, B became an organelle in the A. Now because you have organelles in the A became a eukaryotic cell. Is it clear? Yes, true nucleus, Kavita, true. But eukaryotic cell is not only about having a true nucleus, it will also have organelles. That is the main difference between the two. Is it clear? Not multicellular. Okay. Mandeep, it is not multicellular, it is unicellular only. Why? Because B became an organelle in the A. Did you understand? It lost its identity. It is not like another cell anymore. It became an organelle in the A. Is it clear? So, this is not multicellular. It is unicellular only, but eukaryotic, a cell with organelles. Yes, clear for all of you? Same way, okay, another story. Okay, in this other story, what happened? Okay, there is A. Okay, and this time there is C. A, okay, is very hungry, eating everything. What should I do? Okay, so A ate away C. Gone. C gone inside. Then, okay, A giving shelter. A giving shelter. C doing photosynthesis. C doing photosynthesis for A. And did you guess what is C? Our chloroplast. C became chloroplast and A became a eukaryotic cell. Okay, this is mitochondria and chloroplast. This is how they evolve. Is it clear? Now, check this statement once. Okay, endosymbiotic theory explains evolution of prokaryotic cells from the non-living matter. Is it correct? No, wrong. Endosymbiotic theory explains evolution of eukaryotic cells. Evolution of eukaryotic cells from prokaryotic cells. Agreed? Hello, Ashish Kumar. Okay. So, it will not explain evolution of the prokaryotic cells from the non-living matter. No. It will explain evolution of eukaryotic cells from two prokaryotic cells together became a eukaryote. So, that is wrong statement. Neo-Darwinism explains about formation of organelles like chloroplast, mitochondria. No. Which theory explains? Endosymbiotic theory explains. Endosymbiotic theory explains. Legume plants can convert atmospheric nitrogen to ammonia, nitrogen fixation? No. Okay. Nitrogen fixing bacteria in the legume plants. Nitrogen fixing bacteria in the legume plants will fix the nitrogen. Is it clear? Legume plants nodules, correct. Okay, will fix the nitrogen. Yes, clear for all of you? So, all these three statements are wrong. A very negative question. Okay, no problem. 20th question. Read the statements carefully. Okay. 40 seconds to go. Yes, Ashish Kumar. Correct. Yes, Gaurav. Correct. Okay. The right answer for 20th question is B. Only two statements are correct. Which two statements are correct? 1 and 2 are correct. What is the zombie virus? Interesting. Okay. See, you know, we are facing a huge threat because of the global warming, isn't it? Seasons are changing, unpredictable rainfalls, more floods, okay, more cyclones, and snow is not coming when it is expected, and so all these things are occurring. Global warming, is it clear? Because of that, what is happening? The permafrost, okay, in the polar ice caps melting. Is it clear? Okay. But what is the problem? There is a zombie inside that permafrost. Okay. What is this zombie? Some pathogens, some bacteria, viruses. They are frozen. They are frozen. Okay, last question. Okay. They are frozen for centuries, thousands of years under that permafrost. Is it clear? Okay. Now because they are melting, they are coming out. And they can cause the next pandemics. Did you understand the problem here? Therefore, they are called zombie viruses. Okay. Zombie viruses are the viruses that are frozen in the Arctic permafrost. Yes, true. Scientists are referring to, okay, Methuselah microbes as zombie virus. True. This is the name of it. Nothing to explain, name of it. Okay. Methuselah is one such zombie virus. Okay. Recent discovery revealed that they can be used in green energy production. No, don't worry. That is an irrelevant statement. I tried to make some statement. They have nothing to do with green energy. They are zombies here. Okay. So, they will not help in such good things. Okay. So, these are being studied as oncolytic virus. Okay. What do you mean by oncolytic? Onco is related to cancer. Lytic means killing. Exact word is breaking down. Exact word is breaking down. Okay, no, they don't do good things as zombies. A ghost which is coming out from the Arctic permafrost. Wrong, wrong. Okay, so that is the story of zombie viruses. Okay, one last question. Okay, which among the following is not a diagnostic test of the typhoid? Actually, after this you have one more question also, but you can read it because I kept it in the PPT. It's okay. It is self-explanatory. Read it. My discussion is, the last question is this one. Okay, we are discussing this one as the last question. I kept 22 in the PPT, but we are discussing 21. Read the 22nd one. I hope you are empowered to read and understand the other questions on the other topic with all these sessions that we have done. Is it clear? Okay, now coming back to this. Okay, how can we diagnose typhoid? Okay, culturing the bacterium from the patient's blood? Yes, correct. Okay. See, there is a typhoid patient. Collect the blood. Store it for 1 week. Then what happens? The typhoid bacterium grows. That is nothing but culture. Is it clear? Okay. And then when typhoid bacterium is growing from your blood, what is the meaning? You got infected by typhoid. So we can use it as diagnosis. We can use it as diagnosis. So correct. Next. Polymerase chain reaction, that is PCR. Just like RT-PCR we have seen in the COVID-19. Remember, we can use PCR for any pathogen, not only coronavirus. The only thing is, if I find coronavirus RNA in your blood, it means you got COVID-19. If I find typhoid bacterium DNA in the patient's blood, it means he got typhoid, like that. Okay, Widal test. Okay, this is more important. What the Widal test will do? It will detect antibodies of the it will not detect the typhoid bacteria. It will detect the antibodies for the typhoid bacteria. Is it clear? It will not detect the typhoid bacterium directly, but it will detect the most commonly used. There was a news article recently in the Hindu newspaper about it, if you have at least seen the title of the article. Okay, most commonly used. Why this is most commonly used? Is it because it is very accurate? No, this is very less accurate. This is very less accurate. But, okay, simple test. You don't need sophisticated laboratory. You don't need sophisticated laboratory for this. That is the reason why this is, okay, more commonly used. Next one, enzyme linked immunosorbent assay. This is more famous for HIV, if you listen. That will also detect antibodies. That will also detect antibodies, but this requires sophisticated lab. Okay, remaining all will require a sophisticated laboratory, but Widal test is a simple test that you can even do in the clinic. So therefore, this is the most commonly used, but all of them can diagnose typhoid. Okay, that's all for today. And also that's all for the conquer prelims, and you will conquer the prelims, right? Okay, all the best for your exams. Good night. Take care. Bye-bye.